Mung bean has been used for centuries in Asia as a traditional food and a herbal medicine for inflammatory conditions, and modern research is supporting its anti-inflammatory effects. Two flavonoids in particular, vitexin and isovitexin, appear to be the major contributors to the mung bean's beneficial properties. In one study, the mung bean seed coat was found to contain 96% of the vitexin and the isovitexin, and it accounted for 87% of the free radical neutralizing potential of the bean. In preclinical studies, the vitexin inhibited the production of inflammatory cytokines, and it maintained levels of defenses against oxidative stress. In an animal model of lung injury, isovitexin demonstrated anti-inflammatory effects related to inhibition of cytokines and reduction in oxidative tissue damage. Lab research has found additional compounds called mung bean seed coat saponins, and these also inhibit cytokine expression. One lab trial found the mung bean seed coat powerfully inhibited both a herpes and a respiratory virus to a similar degree as an antiviral drug, while in this case it also induced antiviral cytokines including TNF-alpha and interleukin-6. In a trial in obese mice, the mung bean seed coat significantly reduced the inflammatory cytokine levels. The mung bean sprout extract demonstrated anti-arthritic activity in arthritic rats, and both the mung bean seed coat and the sprout markedly improved glucose tolerance and metabolic health in diabetic mice. In one trial, mice that received the mung bean seed coat were protected from lethal bacterial infection. The survival rate was 70% in the treatment mice versus 29% in the untreated. In a lab component of the study, the mung bean seed coat extract was found to inhibit a protein that mediates lethal systemic inflammation and the inflammatory cytokine interleukin-6, while also demonstrating possible direct antibacterial activity. In a mouse study, the treatment with fermented mung bean preparation protected the mice from cancer while also inhibiting inflammation and stimulating immunity. Black cumin seed oil is an antioxidant and an anti-inflammatory agent, and it's traditionally used to promote digestive, skin, and liver health. Research suggests the black cumin seed oil balances the immune and inflammatory response, and it supports healthy immune system functioning. As we age, there's an increase in the destructive inflammatory pathways, coupled with an impaired immune response, and this is associated with chronic diseases like arthritis, allergies, and cardiovascular disease. Black cumin seed oil and its active components, thymoquinone, are believed to suppress harmful inflammation, while supporting the normal immune response that's required to combat infections and cancer. The black cumin seed oil stimulates the activity of macrophages and helper T cells. In a placebo-controlled trial, 40 women with rheumatoid arthritis took a placebo every day for 1 month, followed by 500 mg Nigella sativa twice a day for 1 month. The arthritis symptoms, including joint pain, swelling and morning stiffness, and disease severity, reportedly decreased after the treatment. In a prospective double-blind study, 66 people with allergic rhinitis were exposed to the black cumin seed oil for 30 days. The symptoms of rhinitis, including congestion, itchy nose and sneezing, decreased after the treatment. The animal research suggests the black seed supplementation is cardioprotective as it reduces lipid oxidation and it promotes healthy cholesterol levels. Reactive oxygen species generated during mitochondrial respiration contribute to inflammation. Aging individuals are especially susceptible to the mitochondria-related oxidative stress, since the mitochondria become increasingly dysfunctional as we age. Taking steps to support the mitochondrial integrity and the efficacy can help to alleviate some of the systemic oxidative and inflammatory burden caused by the poorly functioning mitochondria. Two nutrients, Coq10 and Pqq, are powerful mitochondrial protectants, and studies support their anti-inflammatory role. Pqq is a cofactor for enzymes that are critically important for cellular energy homeostasis and redox balance. Several studies have shown the PQQ exerts a protective effect during circumstantial mitochondrial stress and increased oxidative load. In one study, rats who were given a diet supplemented with PQQ, they displayed greater energy expenditure and remarkably increased mitochondrial density in their liver tissue. PQQ supplemented rats also had lower triglycerides, and their hearts were more protected against the lack of oxygen than the rats that didn't have the PQQ. During periods of limited oxygen supply to the cardiac tissue, a dramatic spike in oxidative stress and subsequent inflammation damages the cells. The findings from this animal model indicate that the PQQ can stave off this inflammatory cell destruction by preserving the mitochondrial efficacy in the adverse conditions. CoQ10 is an indispensable intermediary in mitochondrial ATP production, and studies have shown that CoQ10 levels are low during inflammatory conditions. In one investigation, patients with septic shock were found to have CoQ10 levels substantially lower than the healthy individuals, and among the patients the lower CoQ10 levels were correlated with higher levels of inflammatory mediator called VCAM. In an animal model in which rats were given drinking water with added fructose, an experiment that leads to obesity, diabetes and other inflammatory complications, the CoQ10 supplementation attenuated the inflammatory response by decreasing the hepatic expression of C-reactive protein and other inflammatory mediators. Lab experiments indicate that CoQ10 modulates the expression of several hundred genes, many involved in inflammatory signaling. Of particular significance, one experiment showed that CoQ10, at physiologically relevant concentrations, was able to blunt the induced TNF-alpha by more than 25% via the modulation of NF-kappa B signaling pathway. The role of elevated blood sugar and glycation end products in inhibiting an inflammatory storm has been discussed. Fortunately, in addition to reducing the caloric intake to suppress both the fasting and post-meal glucose concentrations, some natural compounds ameliorate the glycation process, and they might help to rein in the sugar-induced inflammatory cascade. Chief among these anti-glycation nutrients are benfotiamine, a member of the B vitamin family, and carnosine, an amino acid. Benfotiamine has been used to target diabetic complications since the mid-90s. More recent evidence continues to support it as a powerful protector against blood sugar-induced tissue damage. In a clinical trial, 165 subjects with diabetes were randomized to receive benfotiamine at either 300 or 600 milligrams per day, or placebo for 6 weeks. After the intervention period, those who were taking the benfotiamine exhibited improvements in neuropathic pain in a dose-dependent fashion. An animal model found the benfotiamine relieved the neuropathic pain by powerfully suppressing inflammation. Moreover, lab experiments have shown, in addition to blocking the glycation reactions, the benfotiamine may regulate inflammation more directly by modulating the COX and LOX enzyme activity. Carnosine exerts a range of favourable biochemical effects within the body. It powerfully blunts the glycation reactions and it eases oxidative stress. In addition, several experiments have revealed a marked ability of the carnosine to suppress inflammation in various cell types. Unfortunately, carnosine levels decline as much as 63% between the ages of 10 and 70. Furthermore, in patients with type 2 diabetes, skeletal muscle carnosine content is markedly lower than in the healthy control subjects. When the carnosine is administered as a supplement to animals with chemically induced diabetes, it's able to protect the delicate retinal cells from inflammatory complications related to the high blood sugar. Gynostemma pentaphyllum, or jiaozhilang, is used in Asian medicine to treat several health conditions, including dyslipidemia, type 2 diabetes, and inflammation. Its effects are due, at least in part, to its ability to activate a critical enzyme called adenosine monophosphate activated protein kinase, or AMPK. This enzyme affects glucose metabolism and fat storage, and it's been called a metabolic master switch because it controls numerous pathways related to extracting energy from food and storing and distributing that energy throughout the body. Being overweight has a significant effect on AMPK activation and chronic inflammation. It suppresses the AMPK activation, leading to abdominal fat deposits, which in turn activate systemic inflammation. At the same time, inflammation itself suppresses AMPK activation, creating a vicious circle. Greater AMPK activation contributes to weight loss, and it can suppress inflammation. In addition, increased AMPK activation is associated with reduced liver fat accumulation, another source of inflammatory chemicals. Evidence demonstrates the anti-inflammatory effect of the gynostemma. In one lab study, the researchers found the extracts of the herb significantly inhibited several inflammatory chemicals, including TNF-alpha, interleukin-6, and Coq10 mRNA. Another study in 24 patients with type 2 diabetes found drinking a tea made with the herb for 12 weeks significantly reduced the insulin resistance, which is a key contributor to systemic inflammation. Hesperidin and related flavonoids are found in a variety of plants, but especially in citrus fruits, and particularly the peel. These compounds are powerful free radical scavengers, and they've demonstrated anti-inflammatory, insulin sensitizing, and lipid lowering activity. Findings from animal and in vitro research suggest that hesperidin's positive effect on the blood sugar and the lipid levels might be related in part to its activation of the AMP activated protein kinase, or the AMPK pathway. Accumulating evidence suggests that hesperidin might help to prevent and treat a number of chronic diseases associated with aging. Hesperidin might protect against diabetes and its complications, partially through its activation of the AMPK pathway. Coincidentally, metformin, a leading diabetes medication, also activates the AMPK pathway. In a 6-week randomized control trial on 24 diabetic participants, supplementing with 500 mg of hesperidin per day improved the glycemic control, increased the total antioxidant capacity, and reduced the oxidative stress and DNA injury. Using urinary hesperidin as a marker of the dietary hesperidin, another group of researchers found the highest levels of the hesperidin intake had a 32% lower risk of developing diabetes over 4.5 years compared to those with the lowest intake. In a randomized control trial, 24 adults with metabolic syndrome were treated with 500 mg of hesperidin per day or placebo for 3 weeks. After a washout period, the trial was repeated with the hesperidin and placebo assessment reversed. The hesperidin treatment improved endothelial function, suggesting it might be one important mechanism behind its benefit to the cardiovascular system. Hesperidin supplementation also led to a 33% reduction in the median levels of the inflammatory marker C-reactive protein, as well as a significant decrease in levels of total cholesterol, apolipoprotein B, and a marker of vascular inflammation, relative to the placebo. In another randomized control trial in overweight adults with evidence of pre-existing vascular dysfunction, 450 mg per day of hesperidin supplement for 6 weeks resulted in lower blood pressure and a decrease in the markers of vascular inflammation. Another controlled clinical trial included 75 heart attack patients who were randomly assigned to receive 600 mg of hesperidin per day or placebo for 4 weeks. Those taking the hesperidin had significant improvements in levels of high-density lipoprotein, or good cholesterol, and markers of vascular inflammation and fatty acid and glucose metabolism. For more on inflammation, or herbs, supplements, and natural treatment plans, check out my website.